Good afternoon, everyone. Welcome to our research colloquium of January 2021. So today we invite three lecturers from FEDB to share about their recent studies. First of all, let's welcome Dr. Nur Ilya Farhana from Department of Civil Engineering. Her research title is Static Bending Performance of Mengkulang and Kasai LVL beam in structural size.
Yes, thank you, Mr. Uncle Meng. First of all, I'm very sorry that today I have very limited time as today is my very first day of my industrial attachment. Hence, I have a pre-recorded video for my presentation today. Should you have any questions or interested with my research area, you can contact me through my official UCSI email address. So now I'm going to play my presentation video. Thank you. Hi and good evening. My name is Nur Ilya Farhana from Civil Engineering Department. Today I would like to share my paper titled as Static Bending Performance of Mengkulang and Kasai LVL beam in structural size. This is my student's final year project, a collaboration with Faculty of Civil Engineering, UiTM Shah Alam. So this is my presentation outline today, consists of introduction, methodology and experimental setup, result and discussion and conclusion. Production, concrete and steel are the most common structural materials used in construction industry, but actually timber is the oldest and the primary materials used in this industry before these two. Timber is environmental friendly for sure, durable, and its renewable quality maintain its property as a construction materials to build a structure and also as a finishing as well. But there is limitation on usage of timber as a building materials because of the natural properties and its characteristic itself. Timber is sensitive towards different external conditions, highly variable properties and so on. Due to this issue, we need to find and explore alternative way to make sure we can keep using these materials, but we need to improve the properties or modify the properties. It's either by using or by applying any kind of treatments, either chemical or the natural one, or we can go for engineered wood product, EWP, which in this study we use LVL, laminated veneer lumber. Okay, but there is limited data on the performance of LVL in terms of bending, which is in the loading direction of edgewise, and also we are going to explore the different grain direction for both parallel and perpendicular. Figure 1 shows the laminated veneer lumber, LVL, one of the engineered wood product that I mentioned in the previous slide. As you can see here, there is one beam, timber beam, with multiple layer of veneer. Veneer is actually the very fine and thin slices of timber that you can see here that we actually cut and peeling from the actual log, and then we laminated with glue one by one slices to become a beam like this. So this is the objective of this study, to determine the static performance in terms of bending, where we are going to apply the load of the bending load on top of the edgewise side, means on top of the layers of veneers, not on top of the surface of veneers, but on the layers, the side that I show you in the figures. And then we are going to consider the bending load on the both grain direction as well, in terms of parallel direction and perpendicular direction. And there are two types of timbers species were used in this study, where we consider to use the Malaysian hardwood timbers named as Mengkulang and Kasai. Methodology and experimental setup. The test conducted is 4-point bending test, and then we use standards BSEN 408 2010 plus A1 2012 as a reference, where this one is meant for the engineered wood product to determine the physical and mechanical properties of the engineered wood product. And then the loading direction and grain direction, as I mentioned before, we are going to consider edgewise and parallel and perpendicular grain direction. And there are two species of timber used, Mengkulang and Kasai, and these two It's actually for Kasai come from strength group 4 and Mengkulang come from strength group 5, and the timber sample size is 60 mm times 50 mm times 1080 mm in terms of depth times thickness times length. Figure 2 shows the 4-point bending test setup based on the BSEN, the standard mentioned in the previous slide, and the test was conducted at the Structural Heavy Lab UITM. A stand was installed on the sample during the testing to make sure to place the LVDT in order to determine the local deflection. And then the DT3, DT4 were placed below the loading to monitor the uniformity of the loading operation. And DT1 was placed at the center bottom of the sample. This one is to obtain the global deflection reading. And then the DT2 placed at the stand screw at sample's neutral axis. This one is to obtain the local deflection reading. During the test, the data to be recorded were the deflection data through the LVDTs mentioned, and the load data through the 250 kilonewton load cell mounted on the actuator. And then based on the standard, the maximum load must be achieved within 300 seconds. Where the constant recording of the load reading was done digitally through the system until the sample was totally filled. Result and discussion. Oven dry method was used to determine the moisture content, and for density, the density of the samples were determined using measurement and weighing method. All of the mentioned properties is shown in the table 1. For the double line is for the grain parallel to the span, and for the upside down T is for grain perpendicular to the span. From this table, it is observed that all of the LVL samples produced from Kasai possess higher density than Mengkulang. This is as expected, would possess higher strength than the other species due to its high density, as the timber sample strength is commonly assumed to be linear to its density. It's also noted that in the table, the moisture content of the sample were kept below 19%, means that all of these LVR sample were all seasoned or dry and not in the green timber condition, which is not in the wet condition. And the highest MOR, modulus of rupture value, is 71.965 Newton per millimeter squared, resulted from the LVL samples of Kasai with grain parallel to span, and the highest local and global modulus of elastic MOE obtained were 1090.935 Newton per millimeter squared and 12043.918 Newton per millimeter squared respectively from similar sample group of Kasai as mentioned, and the lowest MOR value obtained from LVL sample of Mengkulang in grain perpendicular to the span. LVL produced from Kasai had higher strength and MOE values as compared to the Mengkulang LVL under different grain direction, and the Kasai had higher density compared to Mengkulang as well. This statement fulfill the assumption that the higher density yield a higher bending strength. The grain direction also affected the load versus displacement curve of the samples in bending tests. Figure 3 and Figure 4 show the effect of grain direction on the static bending performance of different timber species. Figure 3 for Kasai and Figure 4 for Mengkulang. In both figures, steeper graph in parallel grain samples could be observed for both timber species, which indicates that the sample with grain parallel to its span were more rigid than the perpendicular one, and there is similarity in both parallel grain sample graph. The yield strength on the parallel grain sample were achieved earlier than the perpendicular one. the perpendicular sample failed at a greater displacement than the parallel one. The parallel grain sample were able to achieve a higher maximum loading as compared to the perpendicular grain sample, indicating that the parallel grain sample were able to take more load before the failure. The result yielded from this study regarding the effect of grain direction were satisfactory. The result fulfilled the assumption that the slope of grain has significant effect on the mechanical properties of a timber sample, which the strength decreases following the increment of grain deviation. Wood is stronger, timber is stronger with grain parallel to span as compared to perpendicular. This is due to the wood fiber orientation that allows the stress to be resisted by the whole length of the fiber when stress is applied. For the conclusion, based on the lab test, Kasai timber species displayed higher density value than Mengkulang, same as the strength group as well. Kasai is in the higher strength group compared to Mengkulang. The MOR and the MOE values followed the same order as the density as well, proving the linear relationship between bending strength, and the density of a timber materials. This study also concluded that the slope of orientation of the grain has significant influence on the mechanical properties of a timber sample, which the strength decreases following the increment of the grain deviation and vice versa. With that, I end my presentation. Thank you so much.
Thanks, Dr. Farhana. Any question from the audience, you may send an email to Dr. Farhana. So let's proceed with our second presenter. So let's welcome Dr. Ramani Varadharajan from Department of Civil Engineering. She would like to share two topics. The first topic is titled as Shape-Based Intelligent Management of Medical Waste Using Artificial Neural Networks, while the second topic titled as Measures to Prevent Total Colloquium Violations in the Compliant Chlorine Treated Water at the Water Treatment Plant and in Water Distribution Systems.
Hi, good afternoon, everybody. Thanks for arranging this research colloquium. It will be very useful for everybody to collaborate, share our research output, grow in research area further down. Thanks for our faculty research coordinator and the head, and also our student volunteers, lecturers who are all supporting our research work. This paper is presented in conference virtually conducted in Italy. It was a wonderful experience to attend such a virtual conference in Italy last month, December. In between our busy schedule, I managed to attend some sessions and presented my papers. I'm thankful again for UCSI and management, survey, and everybody who have supported this event. Let me start with the first presentation on measures to prevent total coliform violation in the compliant chlorine-treated water at the water treatment plant and in water distribution systems. The meaning of the title is to say we need to quantify the biological population in the water to test whether it is biologically safe or not. It is free from biological pollutant or not. The simplest method with which we adapted is using agar, growing agar in the enzymes. That environment was very helpful and easy to assess this population. With that, we have come out with interesting results, which was not yet published. Maybe after this conference, we are planning to publish the results. Okay, the authors are Ramani and Ong, and Gangadharan and Malik. This gentleman is from Department of Environment. He has worked in the area, environmental field, and also in water quality. That's why I collaborate with him, and he has provided some valuable information. Once everything is settled and if the method is validated, it may be standardized in future. But there are some side validation necessary. I'm still working on that. Once it is completed, the method will be standardized for further use. My presentation will follow this title, Background studies, state of art, methodology, results and outcome, conclusion and references. Background. Water is essential. All of you know that. Today, whenever I attend the seminar, I ask whether you are testing for COVID or not, including COVID. In addition to more pathogens and biological population water, you should also check for COVID and different types of muted virus, not only one virus, SARS and its popular other hybrid versions. You need to be checked before we use the water, and also used water, wastewater also very much drawing our attention to test and then standardize their level before they discharge back to the river. Right. Coming to this one. Okay. Coliform bacteria are the microorganisms which are present in the environment and feces of human beings, right? And this bacteria is used as a indicator organism, and it is not a disease-causing organism, so it cannot be called as pathogens, but it will indicate the presence of other bacteria and viruses and other microorganisms present in water by means of simple microscopic testing. Coliform is easier, cheaper to be tested compared to test all possible pathogens, and is not time consuming, very easy and less expensive. But the medium we bought is very expensive for testing. Other than that, the time consumption and results is very fast. Nowadays, on COVID-19, how many hours we test? How many hours needed for getting result? Anybody in the group? How many hours you have to wait for getting the results of COVID positive or negative? How many hours needed? No idea. Minimum hours?
No idea.
14 hours. Okay. 14 or 20 hours, like 14 to 20 hours, and collection of results they ask us next day. So 14 to 20 hours you need to wait to get the results. It's not on spot, right? It is not on situ or in situ, whatever the name you use, on site. It's not immediate. You need to wait for hours to check, and then by the time you finish and you get the results, you are still prone to get the virus, isn't it? It's not that I'm tested, I'm positive. When you go out, mix with the market, go to market or go to some shopping complex, you are prone to get again. Chances, right? Become positive or negative again. That is how it is very crucial and complex to test the presence of biological things or pathogens in water or any air or whatever the media we are using. Students can explore on this area how to minimize the hours, how to many people, many eminent people, the funding agencies starting from Bill Clinton, Melinda Clinton, UU projects, Asian or Asia Pacific projects, many funding development agencies, they are working on this to make it very quick and very fast in getting the results. One of them is this coliform. But the coliform is a very good testing, but it cannot indicate all the bacteria and virus there. To some extent, like I have listed here, we have found out some of the viruses present in the bacteria present in the water. I will give you the result at the end of the presentation. So it is necessary to have in situ testing, quick testing for biological contamination. The existence of pathogens will result in violation of water quality with the symptoms ranging from no ill effects to diarrhea. The symptoms can reveal the presence, but the symptoms are also not always serious, right? So maybe starting from small sickness to diarrhea, vomiting, or body pain, and so on, right? We have chosen the site in the municipal supply of water at the University of Nottingham. This is a student project, FIP project, not FIP project, it's a summer student project. He has worked for 3 months and produced the results around 4th month. It was completed by 2000, I forgot the year, I think in 2018, they completed the results. Methodology. Water samples are collected at the different outlets. Actually, the question was, the research question, if you talk about, we started thinking about the proliferation of this coliform way back in 2003. University, when I was working in University Malaya, people from Serang, Shabas, they approach us and they say they found this type of coliform populations in and then some kind of contaminations starting from the storage. After treatment, after chlorination, disinfection, they use all types. If you go to Selangor water treatment plant, you can see all types, conventional to advanced to membrane technology they are using. In spite of that, they have the population still contaminated. Once it reaches the storage tanks, we cannot identify where the population is, where the problem is, whether at the storage tank or at the distribution or at the main line or at the discharge point. It was very challenging to find the cause and also the effect. Effect also keep changing. Um, we in in addition to this, 2003 was the number one, and the very first they realized this effect, this presence of coliform in the water that is causing illness to the users and also difficult for identifying its uh, location. Once you identify, you can find, you can try to eliminate, right, try to rectify, but it was very difficult to identify itself. Then it was moved to another. After a few years, again the problem of sedimentation, problem of filtration add to this effect. They have caused more dangerous issue, to the extent that it become catastrophic to attend or standardize, come out with a standard testing and results to get proper model or equation to solve this problem of biological testing. So the one of the just we started with the simple testing to see whether it's really present or not, or which part. For that we do sampling, we do some sampling at source, at sampling at the lines, and also at the discharge points. We selected a few points to collect water intermittently, like morning, before sunrise, after sunrise, like around 9 o'clock, afternoon, full sunlight, evening and night. Like that, the sample was tested at different temperatures and different timings. Membrane filtration was used. These are all the facilities available at Nottingham, so we are able to do it, but very expensive. One test itself costs 10,000 about. If you purchase, it will be about 10,000 one type of testing itself. But we are able managed to do this because that was the time the microbiology lab was developed. So we started using this auger. We developed the colonies and then different. We use this Makoni auger with this streak plate techniques. The how it is growing, how it is developed is written here. Osin blue EMB auger in the medium of the medium is used to find the gram-negative bacilli and eccentric bacilli and other types of microorganisms present in it. This was made roughly around 36 grams of agar, which was mixed with 1 liter of water and finally diluted to 1000 ml. Okay, this is the preparation method we use. We follow the standard preparation method, conventional one. We didn't change any of them because our intention was to find out whether there is a population, how much is it, what is the MPN index, and so on. In the beginning, no, according to the standard, before we do tests and come out with the result according to the standards. It varies from region to region. Australian standards say something, Canadian, UK, Malaysian, European standards, they are a bit varying, but in all we can say you should be 0 count in per 100 ml. That's the basic criteria we have to follow. But if you see the results, it was not so. Right. And then these are the temperatures maintained to grow the the auger and the medium is developed like this. Okay. Sample collection. As I said before, we wanted to have testing at different points to identify the location of this population. About 400 to 450 ml water sample was collected in 600 ml bottles for testing, proper incubation, proper handling, and proper cooling effect. Everything was maintained, and the room was very clean. The student will go, even now, rarely I go. The student was very careful, and he was the only person admitted into the room to check every day or replace or place the materials there. Duplication of samples were done with the 100 ml samples per test. So based on the medium created, we are able to do this many tests. Source and method. The sample was taken from 3 main sources, which are the water supply inlet, potable source, and storage tank walls. So we are receiving water, and it is stored at the main tanks in the main water tanks in the university. So that is one point, and then along the line, and then at the tap. Main points, but we have samples and different multiple samples are also taken. And the simple formula used for this one. Later we come out with a model that for that we undertaken another step. It was followed by other students to achieve the model at the end. But in the beginning we follow this simple method of ambient count. So per 100 ml, how much is the value? The name of the test is called swab testing. The inoculating needle was sterilized on. All these things are the chemical oriented things that you need to follow the procedure, a standardization procedure of the samples. 37 to 48 37 degree for 48 hours. Actually 48 hours we managed to for the first few samples, but not for all. But in the beginning itself we got some differences and some significant results in the testing. We followed, and at the end of fourth month he started writing his report and the results. We got some significant results at the time. Physical factors which are hindering the growth of the bacteria in the municipal water supply are physically we found, and we identified based on the site visit, based on follow-up over the months. We found that during sample collection, sediment was found to be deposited beneath the reservoir tank, which creates an environment for microbial contamination. So cleanliness and the drainage system, right? One of the common reason is the drainage system, cleanliness. It's only assumptions, the observations that we made at the site. We need to really evaluate and check whether this is the only reason or other reasons present. A duct well found to be attached at the inlet of the main water supply, which is functioning as a reserved or emergency water supply. Okay, this duct well found to be the source for coliform violation occurrence in the inlet of the main water supply. Unused water. If you leave the water for one day, the next day it becomes urine already. It smells like urine, isn't it? To that extent, it will get affected, prone to get affected by bacteria and virus around. Here, a duct wall which is unused, right? Unused, and there is a chance that it's a duct well of small, about 6 meter only, very shallow well. So there is a chance that it might be affecting. This might be the reason for contamination of the water supply line beside or along the pathway. Wet basin and wet latrine area creates an avid environment for bacterial growth, and the survival capacity is very high in untreated water environment. Yeah, there is also other type of further down. If you see the supply line, there are some catchments, right? The catchments and where people are using for their excretion, for their disposal, everything. That is also the chances of. This only for the results of the site selected. Okay, based on the survey, based on the results we got, we can interpret this way. Wet basins and wet latrines areas surrounding the catchment also one of the reason. Okay, these are the results. This is the location of the campus, and this is the medium EMB agar after being poured into the petri dish, and I will show the pictures one by one. This is the swapping method. First 4 hours is like this, and second line zigzag line, slowly indicating the presence of bacterial growth. And this one important, horizontal and vertical distance measurement. We follow this analytical method as well by following the microscopic observation. And The results are like this. You are seeing the photograph like this one. You can see the same thing. We wanted to mark so that we can follow the other directions. That's why we mark over the plate as well. The covered petri dish. E. coli, Escherichia and EMB agar. You can identify based on the appearance of non-coliform bacteria on the membrane filter. The different color indicates the different types of bacilli and different types of coliform present in the water. Okay. Presence of this one of the type that we found is Enterobacter aerogenes bacterium in McConley agar. This is the using McConley agar. This one is Newton agar, right? We also tested with other type of agar. Other photographs also can. This one is clear. McConley, this one is Newton. CNO is a Newton agar. You can see the differences. Appearance under McConley agar and Newton agar. This is further down. You can see the difference, changes in color, changes in this medium. Also, there is some traces here. The traces are seen here after 48 hours. Okay. Quantitatively, we can say the population like this. Contamination is this level. When you plot between the positioning the sampling, and then the geometric mean count of the coliform per 100 ml is like this 40, and see this very over them. We take the source and this is the discharge point until the discharge point. The count is varying. Okay. Same goes to the we tested for there are lot of A, B, C, D, E block. Each block along the from the source the tank which is available near the entrance of the university until the block E where the civil engineering department is located. We tested one by one. Okay. These are the results. The morning, evening, the one I told you. Intermittent samples are being tested and plotted like this. Distance from the water supply. This one is based on the distance from the water supply. We wanted to check whether the lead time, distance. Distance is the main reason for growth of the bacteria. One is the time of sampling. Another one is the distance. Over the distance also we plotted and checked. And then from the inlet for different sample, all the repeating same. We have that's why I said for all the blocks up to E block, number of samples have been collected over 3 months time, and every day 5 records, minimum of 5 records have been collected. The student will go on almost every day he will replace and he take back the results he observed through microscope recorded. So it was about 1 month, I think nearly 1 month he was doing all the collection and recording of these results. Within that itself we found Enterobacter. This is type of virus was found. I don't know whether the photograph. I will bring the photo. For now, these results we found. The figure 4. Figure 4. Sorry. Figure 4. You can see the structure. The structure through microscope. If you see, you will find out the type of bacteria. We also refer to some micro. It was the very first time we are doing deep into the microbiological study, because we know only bacteria, virus, and fungi, flora, fauna, but it is deep into the structure of the different types of bacteria and virus and its family. So we referred to some texts and publications, and we found that this type of bacteria is Enterobacter aerogenes, And then Staphylococcus epidemic is also found. 6.5. Figure 5. You can see the lines here, the change in color. And Klebsiella virus. Figure 6. Klebsiella also found. These are the effects. Why we bother about causing illness to death, possibility of death, because we don't have vaccine. If you have vaccine, you have some remedial measures. You have some control over the spread. That's why we have to be bothered and find out the causes, find out the effects, find out the reasons for its proliferation. What will cause the enterobacter will cause, to my knowledge, is urinary tract and respiratory system problem. Staphylococcus may cause joints, catheters, large wounds. Klebsiella, invasions, infection, destructive pneumonia. So all about respiratory. Most of the things will start with respiratory system or intestinal, live in the intestinal to affect our parts of the body or the whole system itself. To find out its source so that we can go for proper treatment and proper distribution of water. And there's a repetition only. Actually, some portion of the money I also spent from MOSTI project at that time, so I need to acknowledge them. This is a summer intern. It was a summer project. Intern. There was an intern appointed during its holiday time, funded by the department. So it was able to handle. We borrowed the laboratory from microbiology and medical science, and we are able to accomplish the test and the results. These are the references. Thank you. Any question?
Thanks, Dr. Ramani. Any question from the audience, please? If no question from the audience, I think Dr. Ramani, you may proceed to your next.
May I take a break? May come after Dr. Karim has presented for the second paper.
Oh, okay. So let's proceed to our next presenter first. Yeah. So let's welcome Dr. Abdulkareem Muyideen Oladimeji from Department of Civil Engineering. So he would like to share about the interval analysis of mode shapes to identify damage in beam structures.
Okay, thank you very much, Mr. Moderator. Good afternoon. My name is Dr. Abdulkareem. I'm also from Civil Engineering, as Dr. Ilya and Dr. Ramani. So I'm here to talk about interval analysis of mode shapes to identify damage in beam structures. My co-authors or my co-researchers are Abidin Ganiu, Olukotsun Nathaniel, Isa Mulum, and Williams Bruno. These other four guys are not members of UCSI. They are in some other universities. So this paper, or will I say this research work, was presented at the International Conference on Mechanical Manufacturing and Plant Engineering in December 2020 in Kuala Lumpur. Actually, this will be the sixth. It was the sixth edition of this conference. Okay, so the abstract of this paper or of our presentation is in structural health monitoring, the use of mode shapes in identifying damage is actually very common, but this identification process using mode shapes actually involves the expansion and reduction of the mode shape data. And during this expansion and reduction of this mode shape data, some information are being lost during these processes. So when some of this information are being lost, there is actually a probability or a possibility of having an inadequate, or will I say a wrong damage identification. So therefore, in this study, we presented a simple and fast damage identification method that uses motif of beam structures. So the methodology involves using the interval analysis. It's actually a mathematical method. So we use interval analysis to establish the interval situation of each of the sections in of section of the beam using the mode shapes. And when this is being done, we actually use it to estimate what we call PODE, that is possibility of damage existence. So with this PODE, or with this method, we are also being able to estimate the mode shape increment, which is actually defined as the increment in the mode shape values of the undamaged and the damaged beam structure. So with this, we are able to also establish what we call the damage measure index, which is a product of the PODE and the MSI. The MSI is the mode shape increment, and this is actually being done using numerical model of a simple supported beam using any. Okay, we actually use finite element software SAP 2000, but it can be done using any other finite element analysis software, which can actually do model analysis. Okay, so for our introduction, as we all know, civil engineering structures actually face several extreme conditions such as hurricane, earthquake, overloading, etc., and whatever. So based on this, one of the principal methods that actually been used over the years in assessing structures, be it building, be it bridges, be it dams, be it any kind of structure, is actually the use of visual inspection. And as you know, visual inspection comes with so many disadvantages, or like so many drawbacks. Number one major drawback is, just take the second Finite bridge as an example. How do you visually inspect that kind of big and complex structure? It's going to be time wasting, it's going to be very cumbersome, and you might not even have access to some places. So, based on this, researchers actually came up with a structural health monitoring method called vibration based damage identification, whereby the acceleration responses or the um, displacement responses or the velocity responses are being measured. And when these are being measured, these are being processed using um, other will I say other methods such as wavelet transform, genetic algorithm, ANN, artificial neural network, or frequency response functions, or change in natural frequency. Or we could actually use the mode shape changes. So, and the pioneer of, will I say, this research area was actually these two called Adams and Cowley. You know, they actually used the pioneering vibration-based damage identification method, which is the natural frequency change. But however, these natural frequency changes are actually susceptible to sensitivity to uncertainties, which is noise. And noise could be as a result of temperature change or whatever, or humidity. And also, it's also natural frequency change is inadequate for damage identification, and it's unable to locate damage in symmetrical structures. For example, like a simply supported beam cannot use this natural frequency method to identify damage in such structure. And also, we also say sensitivity to environmental effects. So, as I mentioned earlier, that temperature, humidity actually affects the natural frequencies of of a structure. So, based on this, another method was now being called upon, or will I say another method was established, which is actually the mode shapes study or mode shape method. So studies on this involves PANDI, as you could see over here, good damage localization index. We have well, Wesolowski actually used what we call mode shape curvature of a beam to identify damage. We have Uzevsky and co. actually also identified damage in plates using the mode shape, and we have O'Brien, Zhao. They also actually use, and also Abdulkareem over here. This is me. Actually use mode shape difference in identifying um, damage in different kind of structures, from bridges, see from bridges to beams to cantilever beams to different kind of structures. So, but as we said in the in the abstract part, all these methods that or all these studies that I've actually explained over here actually involves reduction or expansion of these motifs, and with this processing of the motif itself, it actually leads to the loss of some vital information, which may lead to inaccurate damage identification. So based on that, the aim of this study is, as I said earlier, simple approach to detect and locate damage. So the SAT method, which is used, is mode shape. The methodology involves interval analysis of the mode shape to detect and identify damage. And what mode shape are we using, or did we use? We use the fundamental mode shape, which is the first mode shape. And the damage location actually involves different locations. So a little on interval analysis. This might be maybe a little bit above um, your understanding, especially if you are not in this vibration-based uh, damage identification um, knowledge. So this is what we call interval situation of the mode shape. We have the lower bound and we have the upper bound, and over here too we have the lower bound and upper bounds, and we have A, B, C, D. But as we could see over here, this is the shaded one is for the damage, the unshaded one is for the is for the um is for the undamaged. Okay, so we say when the POD is zero, that is when the PODE is zero, you could see when the PODE is zero, then the, or will I say when the interval situation is like this, then the PODE is what is zero. Okay, for B, when the interval situation is like this, then the possibility of damage existence, that is one. That means that damage exists. Okay, then for this one, you could say that there is an overlap between the undamaged and also the damage. It then states that the possibility of damage existence between when we have this interval situation like this is between 0 and 1. And also for this last one, when we have any interval situation such as this, then the POD is 0. Okay, so A and D are actually 0, B is 1, and C is between 0 and 1. So next, we create a finite element model. We use SAP 2000. We have a beam, which is 550 by 10 by 1 meter. The modulus of elasticity is 200 gigapascal, Poisson ratio 0.3, steel density 7 1,850 kilograms per meter cube. And then the support condition states that both ends of the beam are fixed. So this beam with this dimension has a meshing of 1,000 elements, and with this 1,000 elements, each element is 50 by 10 by 1 meter. Then based on also this, we also then have segments. The beam is being segmented into into 10. Okay, then we have our damage location is at what? Is at element 35, which is in segment um, 4. So the damage shows that this element was changed to what? 10 by 10 by 1. So rather than from 50 by 10 to 1, is being changed from 10 to 10 by 1. So based on that, we have we have the simple sub the beam with two side being fixed, and also we have the what's the name? The segment of the beam over here, and we could see each of the segments 1 to 10, and we could see where the damage is being imposed. If you look at here, you could see that this element has been reduced. So the next thing we have on this figure 4 is actually the mode shape. So this is the first mode. This is the second mode. So this is just actually to show how the vibration responses of the beam is or are. So over here we have the natural frequencies. We have first frequency, second frequency, and third frequency. So and over here we have the undamaged and damaged. But when you look at this, the damage and the undamaged, you could see that the change in frequency is quite small. For the first natural frequency, it's actually decreased from 51.85 to 51.58. So that's less than 0.3 hertz difference. And also we have over here 142.81 decreased to 139.91, and over here we have 225.33 decreased to 223.79. So this decrement is just probably about 1% decrement, which is too small for damage identification. And these are some of the um, drawbacks or disadvantages of using change in natural frequency. So now we use interval situations on the fundamental mode shape, and we are able to establish the PODE and the DMI of uh, this of this of this structure. I mean, so over here we have the segment numbers 1 to 10, and we have the PODE and DMI. So based on this, we are able to actually see the POD okay, over here we could see the PODE of segment 1, 2, 3, 4, 5 to 10. From this table, we could see that the PODE of segment 3 and 4 are actually 100. That's 100%. That is, the PODE is 1. But however, we know that our damage is where it's at segment 4. But when you look at this damage, you'll see that it is very close to segment 3, and that is why we have a PODA that is quite high over here. But further analysis using to establish the damage measure index, that's the DMI, we could see that the DMI value for segment 4 is 2.051, and that of segment 1 is 1.045. So having a plot of the DMI, we could see that this segment 4 that is damaged is actually uh, more pronounced than segment 3, which produce a falsely damaged identification. So based on this, we could say that a raw usage of the mode shape using the interval analysis method could actually provide a would I say a proud information or an information to guide to be able to identify damage, even if a more accurate and a more complex method will be used for higher accuracy. So the summary of this actually shows that the PODA obtained from the interval analysis provides an interval situation that provided sketchy damage information. See, because we could have a falsely detected one, though the DMI value is quite low, but at least we could have an idea of where to concentrate our efforts at. So further analysis by obtaining the DMI and the POD provides better and more accurate damage information. The proposed method is simple, fast, and it provides a relatively accurate results. So, any questions from the audience?
Thanks, Dr. Abdulkareem. Any question from the audience, please? If no question from the audience, then thank you, Dr. Abdulkareem. So let's proceed to the last topic presented by Dr. Ramani, titled as the Shape-Based Intelligent Management of Medical Waste using Artificial Neural Network.
Hi, good afternoon again. Thanks for giving me time to present next time, following Dr. Karim. Thanks, Dr. Karim and Dr. Ilya for fine presentation. I was unable to ask questions as I was busy with other materials. Sorry. Okay, we have time to discuss further. Right. My second paper is on shape-based intelligent method of medical waste using artificial neural network. Can you hear me?
Yes, we can hear you.
The authors are myself, Ramani Bai, Abdullah Mazli. This gentleman is also from Department of Environment, and Dr. Zaima, and then Gangadharan. Contents of presentation, background study, state of art, the same titles I have here: methodology, results and outcome, conclusion and references. Automated medical segregation using ANN, artificial neural network. If you are a user of artificial neural network, I don't have to talk much, but I have a quick meaning of what is artificial neural network I can give you. This is to determine the effectiveness of ANNs in recognizing the classifying different medical waste items from images and video. Right. The research question, starting from research question, I want to present. Way back in 2003, again 2003, around 4 or 3, I was attending one conference, waste management conference in Malaysia, in KL. I think PWT was somewhere I attended. There were problems of medical waste segregation they talked about. Like these students, you also, when you attend conferences, they will talk about unsolved problems, million-dollar questions, trillion-dollar questions, which are all the area you have to start working on, and will have a good scope for your research in future. That's how I started thinking of why don't we work on this medical waste segregation and come out with the best model for automation of this segregation. When I review on the papers and the journals, letter to editors, patents, trademarks, I found there is none or just a model, nothing implemented. For your information, even developed countries like Canada have photographs. I have photographs, many photographs. Chicago, many developed countries and cities. Still now, I don't know the current, very recent status, but after this, there are few models and prototypes they develop. I have seen them, but I have no update as on today. But in way back last decade, it was the situation that human being was used to separate. Even in Chicago hospital, human being was employed to separate the tissues, blood, the hand, the butt hand, then the teeth, broken teeth, the utensils, the knife, everything. So it is to that extent it is injurious, hazardous. You can put any word to that to say the seriousness and hazardness involved. Hence, an automated system is important to segregate the waste for the human being, for medical outlets, and for protecting the medical people, medical officials. That was the starting of my research work. I applied for funding. Fortunately, I got through MOSTI at that time. And the determination. So we started with very basic technology, neural network, which has all of you, most of you know, it's robust in classifying, pattern recognition, classification, segregation, and so on. We use this soft computing technique, intelligent method, and that is not the end. It was not successful really. We had 3 stages: neural network, neurophysiologic, and neurophysiologic. We were successful when we did the hybrid one. It was very successful even to the 100%. You may not believe, some of them are being 100%. Error is 0.0002, 0.2, not 0.02, 0.03, and so on. 99.9897 was the result. To that extent, the programmer was, it is the success of the programmer, I tell you. For this project, there were 1 postdoc, 2 PhD students, 1 civil engineering, 1 computer engineering, who was the one doing programming for the neural network. So computer engineer were employed to do this project. It was interesting work at that time. He has, I mean, it's not one PhD student, there were 2 PhD students. One was doing on ANN, and the one was physiology, Iranian. He was successfully completed. We still communicate with each other, but no time for publication. Maybe soon we will publish all this work. It will be very helpful. Started with ANN, neural network. We tried with some of the models and methods, starting from very basic Levenberg marker, and then ANN, backpropagation, the RNN. We can see, I will present the results, some of the results. To determine the effectiveness, first we wanted to test whether the ANN is suitable or not, because the medical waste, if you consider, there are different types. Even if you see the classification and the scheduled waste, I think the scheduled waste have been updated in Malaysia, the environmental department. They updated in 2000, around 2000 they updated the codes, everything, but still the medical waste have not been properly classified, properly handled at that time, and even until a few years back, right? I'm so sorry again, I have no idea of today. I need to work on that. If I have good student, we will work on that to know much and then come up with a product, real product trademark. We end up with a prototype only. Okay, so to say that whether this is started with the neural network technologies, and we work on it to develop instrument that can help automation of the process. Coming to the government, so there is no proper classification. This project also helped with the postdoc who was an environmental scholar. She has come out with a proper classification. We can even recommend to government departments to standardize these types of classification. Ongoing, you can say. The idea is to come out with a commercially productive machine or a robotic arm to help us. Okay, inspired by the structure of the human, all of you know that. I mean, today I think all branches of engineering have to know, and they're using it in their project or in any other investigative things. It's inspiration how human brain mimic the human brain, how human brain assimilate the information and give out the results for the given input. The same pattern is same way of programming is done in computer to make the computer think like human being. That is what artificial neural network. You are creating an artificial network system in a computer to think and give you the answer, not like crisp mathematics which says 2 plus 3 is equal to 5, right? If we go to fuzzy, 2 plus 3 is not 5, it is 5.0 to 5.1 to 5.3 until the n number. So to process the fuzzy information to the gray area, we are using these types of ANN. ANN is made of one or more layers, and the structure I will explain. Read for that later on. ANN has a structure, input, output. In between, we have hidden layers. Sometimes without hidden layers or one layer, number of layers. There are different types of modeling you can do it. We started with, where is that? We taken the perceptron, started from perceptron, and then we did for object recognition, started with definite shape, like the syringe, bottles. There are different types of ways, starting from hazard, schedule, chemical, the food. The cafeteria in medical also is a type of waste, right? Food waste, and cotton, syringe, and then the petri dish, the test tubes, scissors. You know, the doctors, when they open the packet, and they use one scissors or one, forgot the name, and they use it, they have to throw. To that extent, they wasted the equipments, they dump the waste and become injurious or hazardous to the disposal personnel or those who are working on the waste disposal. And then we started with the shape recognition. So whichever is having definite shape, unlike the cottons, the different types of pads, right, which is not having definite shape, right? We started with the definite shape, like syringe. We are able to model with the normal ANN neural network, and the texture modeling cannot be implemented. That was successful to using fuzzy logic later on. Come on, testing confirm ANN capable of recognition. Okay. So with that, we found that ANN is capable of recognizing the shape, classifying the shapes. And we use these two for image processing. We use chain codes and centroidal profile. You know, if you're having a definite shape, you can find the centroid, right? We can draw the centroid profile. So we use these two methods. We started with the chain codes, and then we also did using centroidal profile. I think explain. Chain codes, I don't want to give you more explanation on this, but I will give you the results. Okay, this is by the pixel by pixel. It will count, and then it will reach the starting. We have to start and reach the end point until the shape is recognized. There is a number, there is a code for each identity, each object. That's how we divided the classify the samples in simple way. I can explain. The other way of classifying the samples are by drawing centroid profile. If this is the syringe, you can see the centroid profile varying from starting to end. The centroid profile is varying until this point. And then petri dish, this is the centroid profile drawing, the trained one. For training, we use 40 hidden neurons. Training size, set size is 40 images. So one side we use 40 images, but the images most of them we photograph and we purchase to stock images, and then we also go to hospital. One of the collaborators from University Malaya Medical Dentistry. So we could able to go there, collect the data, do the site validation. We did it. We have all videograph. I can show, if time permits, I will show a short videograph of our validation as well. Training algorithm, we use this, started with 11 bug marker, and then time taken is 10 to 15 minutes, very fast, because I used at that time the working station with the high config computers I use for running this program. Time taken increase with larger training set. Of course, if you increase the set of samples, it will. For getting more accurate answer, you need to provide all types of the maximum number of images that is possible and understood by your network. So we did it, and as you increase the time sample, the time taken overnight, that student was very dedicated. I should appreciate him. The electronics student, the first student who worked on the ANN, worked overnight. We many days, we stayed in the university with all the team working overnight, finished, and because the team was verified every milestone. I don't know about the current situation. At that time, mostly come for every milestone. They will knock the door every 6 months. Hey, what is your project? What is your result? They are more keen about getting results than us. Curious. So we had meetings quite often. So we'll be very anxious to get the results in the next day. We have to run the program complete and then produce the results for the reporting. We will be looking at ways to reduce training time without sacrifice. Yeah, we also try to reduce them because if for now we can model and develop and implement at the medical outlet. What if there is a new waste coming in, new modification have to take place? These are all the challenges. For that, you should make your program quicker, effective to admit for new data, and then for making multiples, like whether I have to have this unit every floor or only at the patient room or wards or every level. All these things are questionable. In that case, if your program is efficient, you can make full use of the features. Flowchart on this. Only ANN I'm explaining here, presenting here. Select classes. So the flowchart, step 1 is to select the classes to be trained. We had medical classification about 7 in the beginning, and then we increase the number as we get the samples and we could be successful. We increase the number of classes. Step 2, load the images. So we use the neural network, all MATLAB programming with the image processing tool and neural network. This is the image processing tool used for pre-processing the images, all the errors, all the flaws, what do you call, I forgot the term even. We remove all the black spots, or we fine-tune the images by pre-processing. Select and extract the features and loading images. Right? Combination of neural network and pre-processing has helped us to identify the image and extract the features that can be fed into neural network for its training. So we train with the controlling the parameters, like we adjust the threshold and the hidden number of neurons, hidden layers, train, and the features have been extracted. The screenshot below show the objective. Yeah, here's the result. You can see that number 1 method, chain point method, it was able to identify 38 objects out of 40 fed, and central profile method can identify 39. So a bit improvement over the method that we use. 39 out of 40 were correctly identified. You can see name also, the program we put identification and name, labeling, identification, labeling, pipette, tube, PCR tube, pipette, syringe, petri dish, and so on. So able to identify these objects successfully by using neural network and image processing. The last that for the first milestone, the challenge was identification or recognition of the texture. In the prototype, in the development of prototype, we need to look into light control, angle of the light. Even sometimes the student won't let us to go inside. We have to the data will get changed. So it is not robust at that time using neural network. But when you go to fuzzy logic, it was so useful and way forward for us to model the entire medical waste. That was a good thing I have to mention. Shape recognition is sufficient. Rigid bodies, okay. Rigid bodies like bandages, cotton balls, not rigid bodies. However, in our application, many objects are not rigid. Irregular, spongy type, no shape, like bandages, cotton balls, gloves, tubes, packaging things. Packaging is also sometimes they crush it and put it, and it should be able to identify, right? Your robot should be able to identify. So become a challenge for us. We plan to implement texture recognition and have these two recognition systems complement each other. So what we did, we go for next step, which can complement each other. But all of them, once we get all of them classified by fuzzy, we just ignore this neural network at that time. That's all. Grateful for MOSTI, Malaysian Ministry was funded this project way back in 2012, finished in 2012, 2 years, 10 and 12, 10 to 12. These are the publications. And you have any question? I have a short video. If you want, I can run this video. our validation.
Thanks, Dr. Ramani. Are you going to play the video?
Yeah, give me 1 minute. For the panel.
Unless you give him money, he cannot. This is yeah. No, not plastic. One is because it's made of ceramic.
It's a genuine
Ceramic. Very expensive. We try to suppose things are proper.
At the time, just
You do not know your expenses. So I have to plan for the illumination as well.
okay, thank you, sir.
Thanks, Dr. Ramani. Any question from the audience, please? Okay, so if no question from the audience, I think we end our research colloquium here. Thank you, Dr. Ramani.
Yes.
So yeah, thanks everyone for attendance. And for information, we will have a research colloquium every month. And all students are encouraged to join us also. If you are interested to share about your recent progress, your recent research progress, you may register your title with Dr. Song Wei-Ing. So yeah, that's all for today, and see you on our next research colloquium in February 2021. And before you leave, please help me to fill up the Google form that I have sent in the chat box. It is the attendance list for today. So thank you.